दिल की गहराइयों से स्वागत है आपका बायोमोलिक्यूल्स की इस डिस्कशन सीरीज में। पिछले डिस्कशन सीरीज में हम लोगों ने पॉलीसेकेराइड्स के बारे में डिस्कस किया था। इसमें हम लोग 9.6 या न्यूक्लिक एसिड से चालू करेंगे। तो जल्दी से रिवाइज कीजिए न्यूक्लिक एसिड्स को। चलिए, द फर्स्ट क्वेश्चन, द अदर टाइप ऑफ मैक्रोमोलिक्यूल that one would find in the dash dash insoluble fraction of any living tissue is the nucleic acid. The next, these are dash dash. The next, together with polysaccharides and polypeptides, these comprise the true dash dash fraction of any living tissue or cell. The next, for nucleic acids, the dash dash block is a nucleotide. The next, a nucleotide has three chemically distinct components. One is dash dash compound, the second is dash dash, and a third is dash dash acid or phosphate. The next, as you notice in figure 9.1, the dash dash compounds in nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. The next, adenine and guanine are substituted dash dash while the rest are substituted dash dash. The next, the skeletal dash dash ring is called as purine and pyrimidine respectively. The next, the sugar found in dash dash is either ribose, a monosaccharide pentose, or dash dash deoxyribose. The next, a dash dash acid containing deoxyribose is called dash dash acid, that is DNA, while that which contains ribose is called ribonucleic acid, that is RNA. The next 9.7, structure of proteins. जल्दी से रिवाइज कीजिए 9.7 स्ट्रक्चर्स ऑफ प्रोटीन। फर्स्ट क्वेश्चन, dash dash as mentioned earlier are dash dash polymers containing strings of amino acids. The next, structure of molecules means different thing in different contexts. In inorganic chemistry, the structure invariably refers to the dash dash formulas, example NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecule while representing the structure of the molecule, example dash dash, comma dash dash, etc. The next, physicists conjure up the dash dash dimensional view of molecular structure while biologists describe the protein structure at dash dash levels. The next, the sequence of amino acids, that is the dash dash information in a protein, which is the first amino acid, which is second, and so on, is called the dash dash structure, figure 9.3a of a protein. The next, a protein is imagined as a line, the left end represented by the dash dash amino acid and the right end represented by the dash dash amino acid. Then next, the first A amino acid is called as dash dash amino acid. Then next, the last amino acid is called dash dash amino acid. Then next, a dash dash thread does not exist throughout as an extended rigid rod. The next, the dash dash is folded in the form of a dash dash similar to a revolving dash dash. Then next, of course, only some dash dash of the protein thread are arranged in the form of a helix. Then next, in proteins only right-handed helixes are observed. इस सेंटेंस में एक वर्ड रॉन्ग बोला गया है, फाइंड दैट वर्ड. The next, the dash dash of the protein thread are folded into other forms in what is called the dash dash structure. फिगर 9.4b. जल्दी से माइंड कैप्चर कीजिए इसका. The next, in addition, the dash dash chain is also folded upon itself like a hollow woolen ball, giving rise to the dash dash structure. फिगर 9.4c. जल्दी से माइंड कैप्चर कीजिए इसका. फिगर 9.4c का. Then next, this gives us a dash dash view of a protein. The next, dash dash structure is absolutely necessary for the many biological activity of dash dash. The next, figure 9.3, various levels of proteins. स्ट्रक्चर का माइंड कैप्चर कीजिए. अपनी आंख बंद कीजिए. क्या आपको ए प्राइमरी लेवल ऑफ प्रोटीन स्ट्रक्चर दिख रहा है? बी सेकेंडरी लेवल ऑफ प्रोटीन स्ट्रक्चर. इसमें अल्फा हेलिक्स दिख रहा है. बीटा प्लेटेड शीट. पॉलीपेप्टाइड वर्ड कहाँ लिखा है? सी टर्शरी, जिसमें हाइड्रोजन और डाइसल्फाइड बॉन्ड दिखाया गया है. क्वाटनरी. चलिए हम लोग चलते हैं दिन नेक्स्ट पैराग्राफ की तरफ. जल्दी से रिवाइज कीजिए इसको. चलिए, द फर्स्ट क्वेश्चन, सम प्रोटीन्स आर एन डैश डैश ऑफ more than one polypeptide or dash dash. The next, the manner in which these individual folded dash dash or dash dash are arranged with respect to each other. Example, dash dash of spheres. Spheres arranged one upon each other in the form of a cube or dash dash etc. is the dash dash of a protein, otherwise called the quaternary structure of a protein. फिगर 9.4 डी. जल्दी से इसका माइंड कैप्चर कीजिए. The next, dash dash human hemoglobin consists of dash dash subunits. The next two of these are identical to each other. Hence, two subunits of alpha type and two subunits of dash dash type structure constitute the human hemoglobin, that is HB. चलिए आँख को क्लोज कीजिए। क्या आपको फिगर 9.4 कार्टून शोइंग ए सेकेंडरी स्ट्रक्चर एंड बी टर्सरी स्ट्रक्चर ऑफ प्रोटीन, आँख बंद करके देख पा रहे हैं आप? The next 9.8 नेचर ऑफ बॉन्ड लिंकिंग मोनोमर्स इन अ पॉलीमर। जल्दी से रिवाइज कीजिए इसको। The first question from first line of NCERT. In a dash dash or a protein, amino acids are linked by a dash dash bond which is formed when the dash dash group of one amino acid reacts with the dash dash group of the next amino acid with the elimination of water dash dash, that is, the process is called dehydration. The next, in a dash dash, the individual monosaccharides are linked by a dash dash bond. The next, this bond is also formed by dash dash. The next, this bond is formed between two dash dash atoms of two adjacent monosaccharides. The next, in a dash dash acid, a phosphate dash dash links the dash dash carbon of one sugar of one nucleotide to the dash dash carbon of the sugar of the succeeding nucleotide. The next, the bond between the dash dash and dash dash of sugar is an ester bond. The next, as there is one such ester bond on other side, it is called dash dash bond, figure 9.5. चलिए फिगर 9.5 का माइंड कैप्चर कीजिए। डायग्राम इंडिकेटिंग सेकेंडरी स्ट्रक्चर ऑफ डीएनए, इसको कॉपी पर बनाकर मैच कीजिए एनसीईआरटी से। कॉपी मतलब नोटबुक। The next, dash dash acids exhibit a wide variety of secondary structures. The next, for example, one of the dash dash structures exhibited by DNA is the famous dash dash model. The next, this model says that DNA exists as a dash dash helix. The next, the two dash dash of polynucleotides are dash dash, that is, run in the opposite direction. The next, the backbone is formed by the dash dash sugar chain. The next, the dash dash bases are projected more or less perpendicular to this backbone but dash dash inside. The next, dash dash and dash dash of one strand compulsorily base pairs with dash dash and dash dash respectively on the other strand. The next, there are two hydrogen bonds between A and T and three dash dash bonds between G and C. The next, each dash dash appears like a helical staircase. The next, each step of dash dash is represented by a pair of dash dash. The next, at each dash dash, the strand turns dash dash degree. The next, one full turn of helical strand would involve dash dash steps or dash dash base pairs. The next, attempt drawing a line diagram. The next, the pitch would be dash dash angstrom. The next, the rise per base pair would be dash dash angstrom. The next, this form of DNA with the above mentioned silent feature is called dash dash. The next, in higher classes you will be told that there are more than a dash dash form of DNA named after English alphabet with unique structural feature. You are most welcome in the 9.9 dynamic state of body constituents concept of metabolism. Thank you friends. लाइक शेयर एंड सब्सक्राइब प्लीज़। अरे यार ये फ्रेंड का चैनल है और फ्रेंड से प्लीज़ कहना कोई शोभा नहीं देता, इसलिए लाइक कर दो मेरे यार। हम सब आपस में फ्रेंड हैं, तो प्लीज़ लाइक शेयर एंड सब्सक्राइब। बिना पूछे बूझे हुए जल्दी से ठोको, लाइक ठोक डालो।